welcome back to the lecture series in nanotechnology in agriculture. So, as of now we have talked about iron pyrite and the role of iron pyrite in the evolution, and apart from it we discussed how iron pyrite is synthesized in the lab. And if you follow up the lecture previously, so this is an inert environment where you make polysulfide, and the polysulfide is allowed to react with iron salt, and this whole thing happens in an inert environment, because as soon as iron comes in contact with air, it will get converted into Fe3 plus. Okay, it will get oxidized. So, in order to avoid that oxidation, you have to carry out this reaction in an inert environment. And apart from it, as soon as you synthesize these particles, you have to characterize them. So, if you remember all the characterization tools what we talked about, XPS, which will help you to test what is the oxidation state, then the presence of sulfur and the different form of sulfur it is present, the amount of oxygen in the system, and all other different residual atoms which are present there. Okay. So, most critically it will be these two, detecting iron and sulfur. Then for the structural characterization, one can perform scanning electron microscopy and transmission electron microscopy. In order to understand the depth profile, one can use atomic force microscopy, and in order to understand the crystallinity of the structure or the amorphousness of the structure, one has to do an XRD, X-ray diffraction, in order to figure this out. So, these are the basic parameters what has to be worked out, and of course, the particle size distribution at what range particles are when you are synthesizing in such a situation. Now, from here I told you that there was a seed treatment study. So, it is a 8 years long study which have been published in different form of manuscripts over the years with different crops. So, what we will do, we will go to the very genesis of this study, how it all started. So, since nanotechnology is ushering into the agriculture, so every step we are making, you have to make it with a word of caution that we are not unnecessarily tinkering with nature. As we have already discussed that over last 100 years, several things have changed, and there is a lot of environmental concern which has been raised over the use of excess fertilizers and the contamination of the fertilizer into the water table and so on and so forth. So, the first set of experiment what was done was nano-FeS2 or nano-Pyrite. This in itself is not soluble in water, it is insoluble. So, in other words, when you add pyrite in water, it remains as in suspension. So, you can call it a colloidal suspension. So, essentially it is an aqueous, you can put it as aqueous colloidal suspension. So, now this colloidal suspension does couple of things. We have already talked about the stoichiometric defect, if you remember. So, just to recollect it, FeS2, when we talk about molecule like this, there are certain such sulfides which come with a word called stoichiometric defect. In a stoichiometric defects, what happens? So, our assumption is there is one atom of iron and two atoms of sulfur, but when they form a mass, then there will be few iron pyrite molecules. So, for example, something like this, and each one of these circles are representing a single molecule of FeS2. Okay. So then there will be some molecule where there will be a surface defects and which will make them devoid of sulfur. So in other words, there will be some, there will be very few, but there will be some, and that leads us to write 2 as 2 minus x, and that x is essentially the percentage of defects, and this is what we call as stoichiometric defects. So this stoichiometric defect in FeS2 leads to some unique surface reactions. What happens in this is FeS2 plus water, which is insoluble, it leads to formation of a very trace amount of H2O2. Okay. In other words, water is getting oxidized. This is precisely one of the trace, and this H2O2 further kind of act on the surface and kind of release some kind of Fe2 plus, Fe3 plus and SO4 minus ions, and all these are present in a very trace amounts. So in other words, when we talk about a mixture like this or a colloidal suspension like this, so your predominant compound is FeS2 plus a very trace amount of H2O2, a very trace amount of Fe2 plus, very trace amount of Fe3 plus, and a very trace amount of SO4 minus, and there are protons which are present, and such suspensions are slightly acidic. So in other words, it leads to a very complex milieu of ions out there. Now when the very first experiment was done, it was done in a very simple way. So chickpeas were taken. All of you are aware about chickpeas. Okay, these chickpeas were treated with surface reactions to get rid of any kind of fungus or microbes or something. Okay, and then these chickpeas were divided into two categories. Okay, so one category was kept overnight in deionized water, and the other one was kept in FeS2 suspension with equal amount of water. The red ones are the FeS2. Okay, and here you are having the chickpeas which are sitting there. Next day, after 12 hours of treatment, these were placed on test tubes like this. All of them were placed on test tubes. So initially seeds were taken out, both the cases they were washed and they were placed in test tube having deionized water. So here you are having the test tube. Okay, so now you put a thin films out there. On that film, this is a film you can make with some of those sealing agent or something, a small tube with a small hole, and you place the seed out here like this. Place single seed on each one of these parafilm which you have placed, and on that you are having just simple water. Okay, Now what you are essentially trying to do, you are doing a germination test and you wanted to see the growth in pure water. So what you have done, you have exposed these seeds to an ionic milieu of iron and sulfur and pyrite in a slightly acidic environment for 12 hours, and then you are growing them. So post this experiment was continued for 7 days, and after 7 days the results were evaluated, and all these 7 days they were kept in water. So the early phase they only got a booster of 12-hour boosting of these different ionic milieu. So this was the only nutrient which was given to these seeds, okay? Iron and sulfur. In other words, you have fortified them in iron and sulfur, okay? Now the results were very interesting. So after 7 days, what was observed was, so when they germinate like this, and then they form the root systems. Now the one which were treated were more taller and having a much more well spread out root system. And so this was the basic difference which was observed in these treated seeds. Their germination percentage was higher as compared to control, 0.1. 0.2 was their root growth was significantly more, their shoot growth was significantly more, and the fourth interesting aspect was overall biomass was significantly higher. Apart from it, it was observed upon analysis that iron concentration in both these plants remain more or less constant, okay? But definitely there was a difference in the sulfur concentration. Sulfur was higher on these plants, and apart from it there was a difference in few other elements. But this was the very first clue to tell that here is a seed biostimulant, very simple seed biostimulant, which could increase the germination percentage. And this aspect is very critical for a farmer, because if the germination does not happen right, the crop is standing what we call as the height of the crop. If it is not uniform, the height of the crop or the crop is standing will be very poor. So here is a very interesting chemical tool in the armory which helps in increasing the germination percentage. But when you do these kind of studies, the very next question comes. There are two things which comes. The first question which comes is, could you translate this to the field? Is it translatable? To the field the farmers. Second, when you talk about translatability, what is the cost involved? Second, does this treatment exhibit any undesirable effect? Now, both these questions could be answered by performing two things. First, a field trial, because that's essential to prove that how long this effect remains. Is it only up to germination, or this effect of germination has some kind of a prolonged effect? Okay. Now, apart from it, the next set of analysis is what is the possible mechanism of its action on the seed. Without understanding it, it kind of becomes a black box, what is happening. So, field trial, and next is the mechanistic understanding, at least at the ionic level, and long term genetics study, and minimizing or / optimizing synthesis cost of pyrite. Having said this, let me tell you, there are pyrite mines all over the world, but the problem with pyrite mines are the bulk pyrite, what is obtained, has to be either freed from the contaminants like cadmium, arsenic and all other things, and it has to be either ball milled or using the techniques what you have learned, its size has to be reduced down into the nano dimension. Okay? So, that is an issue which has to be addressed before we kind of get a grip on using the huge amount of pyrite which is available. Now, coming back. So, these are some of the stuff what needed to be answered before this study could be taken to the next level. So, then comes the next set of study what we will be talking about soon is the field trial. So, in order to do the field trial, one has to first of all, those of you who are from agriculture background will realize, you have to do, if possible, the double blind experiments. You have to really divide the field in a random fashion, like you have plots which are obtaining equal amount of all the resources, and then you have only one parameter what you are varying. So, in order to test this technology, the basic conditions which were laid were like in the fields what will be used for the field trial. So, the first you select the crop, okay. Selection of crops. So, the first crop where you can see the profound effect in a short term, spinach. And this experiment was done in IIT Kanpur nursery. Okay. Second thing is, of course, the criteria for selecting spinach is the short term, and second aspect is no fertilizer will be used, because that will make the things way more complex. No fertilizer use, neither in control nor in test. Okay. So these were the basic criteria which was laid before this field trial was initiated. And whenever you do field trial, you should have multiple replications so that you can really compare the data neatly what is happening. Okay. So let's recap what we talked as of now. So we talked about the stoichiometric defect out here, where I told you there are some of these atoms where there is a sulfur is deficient. Then we talked about the aqueous suspension have all these components present, and slowly we will divulge what are the roles of individual components which are present there. And then we talked about the experiments and the key results, where the germination percentage got higher, the root growth goes higher, shoot growth goes higher, and the overall biomass got higher. So the root growth, more root growth has a different significance, and which will be coming while we will be concluding this iron pyrite part. What does that mean? Because that is something really very interesting for nutrient deficient soils. Okay. We will come to this part once we finish these field trials. Okay. On nutrition deficient parts or the soils. Okay. So in the next lecture, we will talk about the detailed field trial using spinach as a test crop. Thank you.